is an assistant professor at the School of Integrative Plant Science, Plant Breeding and Genetics section at Cornell University. If you haven't checked out his background, you could find his details on the Cornell University website. His major interests are bioenergy, increasing understanding of G by E interactions, and international agriculture. So he has a lot of connections with different institutes that work to improve crops in Africa. His lab also does research to develop and implement advanced modeling and informatics. He also has a background in animal breeding as well as human genetics before finally working with plant breeding. So he has a very vast, broad background than most of us are used to. His lab is, like I said, focused on bioenergy, genomic selection, phenomics, and G by E interactions with multiple crops and breeding programs collaborating in Africa and Asia. He also teaches the Advanced Statistics and Design course at Cornell. He also teaches the graduate level dissertation, Evolution of Genetic Modeling. So he has a pretty much very wide background, and we're all very excited and happy to have Dr. Robbins join our webinar. So thank you all again for joining us, and I will let Dr. Robbins go ahead and share his screen.
All right, great. Thank you for that introduction, and nice to meet all of you virtually. So let me go ahead and share. All right, you should see my presentation now. I'm going to go into, are you seeing the presenter view or the notes view?
Yeah, I can see the notes view, so.
All right.
Yeah, perfect.
Should be good now? Okay.
Yeah.
I'm looking at the wrong screens, so you're going to kind of get my side profile if you're looking at the video. But all right. So thanks for the invitation to speak to you today. It's really great to be able to engage in this way. This is a topic that I've never presented on before. How to write an effective abstract, and I must admit, I probably haven't put as much thought into this in the past as I probably should have. So this has been a bit of a learning experience for me as well, and hopefully this proves to be at least as informative for you as it was for me as I kind of went through preparing this presentation. All right. Okay, so I'm just going to rearrange some things on my desktop. All right, so why is the abstract so important? So the key with an abstract is that it's one of the first things. If we're talking about a research paper, it's one of the first things people are going to see with your research paper. And it impacts a lot of things, right? So it's, here I have kind of not a very good search, but kind of a search entered into PubMed over here that's brought back some articles. And so, for starters, the title of your paper or your manuscript. So here I'm talking about a manuscript, not necessarily an abstract that you're submitting to a conference, but certainly when we're talking about, a research paper, your title and your abstract and how you write your title and your abstract is going to have a big impact on where it shows up when someone is actually doing a search. And that's pretty important, right? Because, there's a lot of, research papers, high quality research papers published every year. And, certainly me as a scientist, I don't have time to read every paper that comes out in fields that are related to what I do. And so, I have journal alerts that come into my inbox that are based off of keyword searches. If I'm looking for something specific, I'll go to PubMed or I'll go to Google Scholar. I'll try and enter some searches. I'll see what comes up. But ultimately, what I end up with is going to be some long list of publications, and based off of the title and based off of the abstract, that's really going to determine whether or not I actually read that paper. because the abstract should give me a very clear idea of what that research was, what the results were, and what the conclusions were. And based off of that, I'm going to make a determination of whether or not this is a paper that relates to the work I'm doing. At a research conference, much the same way, right? I'm going to, but before I arrive at that conference, I'm going to look at the conference program. I'm going to search through all of the presentation or all of the abstract titles. I'm going to read through the abstracts, and based off of that, I'm going to decide which, especially in a concurrent session conference, where I'm going to have to move around from session to session. The abstract is what's going to determine what talks I actually show up for, and so it's really important that the abstract, you put a lot of thought into how you write the abstract, because ultimately, as a researcher, we want our research to be viewed and known by the community. And how we put together that abstract is really going to play, a part in, how many people are actually going to read our work. And so it is something that I think people generally undervalue and don't put as much of a thought into as they probably should. All right, so before I get started, as I said, this is not a topic that I've presented on before. The first thing I did is I started to go online and look around and see what was out there. And so here are some online resources. I think there's probably ways that we can make this available to you later on, but here are certainly some of the things that I found online that I found particularly useful for me. And in particular, there's a blog post here, from earlier this year that I found to be quite informative. It's very short. It's kind of to the point. I'd recommend everyone, take a look at this. Maybe, keep the link handy, because every time you write an abstract, this might be something that's worth going back and just taking a quick look through, because I do think. And there's also a presentation associated with this that I haven't listened to, but I imagine it's a pretty good presentation that's probably worth following up with as well. All right, so let's just start with what makes a good abstract, right? So the first thing is it needs to be a standalone summary of the research, right? And so I should be able to read an abstract independent of the paper or independent of any other information that's out there, and I should have a very clear idea of what you did, why you did it, what the results were, and what the conclusions were, right? And so that's a really critical component of an abstract, and I'll get into maybe some common mistakes a little bit later, but that there are these key components of an abstract, right? Which really need to be in every abstract that you have, and you need to have a right balance between how much detail you put into each of these components, because with an abstract, there's always word limits. So every abstract that you write should contain really 4 major elements. One, so the first is the background, right? So what is the subject of the research and why is it important, right? So what were you working on, and why should I care about it? Methods, what was done? Methods is a harder thing, I think, to cover in an abstract, right? Because you need to give the reader an idea of what you did, but obviously you have very limited space to do this, right? So you have to keep it brief, and you really need to focus on just like the key aspects. This is not something that you really want to go into excruciating detail, because if you go into excruciating detail, you're not going to have room for anything else. The results probably is the component of the abstract that you should be spending the most space on. I mean, you obviously have to balance across these 4 components, but it's really important to share what your findings were. You need to focus on the most relevant results. Again, with everything in an abstract, you're not going to be able to give a complete representation of everything that's in the manuscript, but you should stick to the most relevant results. And you also need to be careful to not be very misleading, right? there could be a temptation to only really focus on the positive things that you found, just to kind of highlight what was really some favorable results. What you put in the results section needs to be fairly representative of everything that's going to be in the paper, good or bad. And then the conclusions. Conclusions, you want to keep it brief, but it is important to provide some interpretation of the results in the abstract as well. But again, the real trick with an abstract is just finding that right balance between providing enough detail to make it informative, but keeping it brief and succinct enough to fit within the word limits. And that's not an easy thing to do. All right. Finally, it should be as detailed as possible given the word limits. And again, that's really the challenge, right? No matter where you're submitting an abstract to, there's always going to be some limitation on the length of the abstract, which is going to impact your how you summarize and how much detail you put into the abstract you're presenting. And so I guess to continue on with the theme of genomic selection, which is what I had kind of put in the search engine earlier, I figured what we could do now is we could actually walk through some abstracts, highlight the various components of the abstracts, and kind of discuss, what was done well and what may have not, gone so well in some of these abstracts. And so for the start, I'm gonna focus on a paper that I think anyone that's working with genomic selection and probably just in breeding in general is probably familiar with. This is a paper from 2001 by Munison, Hayes, and Goddard that really sort of launched the field of genomic selection and genomic prediction. I think this paper's been cited something like 5,000 times. I'm not entirely sure, right? But this is, a very big paper in the field that kind of represented a big paradigm shift. So I figured, well, maybe this is a good abstract to start with. Because if you have a paper that has 5,000 citations, you probably did a reasonably good job with your abstract. All right, so let's break this abstract down into the 4 components that we highlighted before. So let's start with the background, right? So background, it should tell us what the subject of the research is and why it's important. So let's look at the background statement here in this abstract. Recent advances in molecular genetic techniques will make dense marker maps available and genotyping many individuals for these markers feasible, right? So this is a great start. It's a bit of a forward-looking statement. It's basically saying we're about to hit a paradigm shift where we can genotype many individuals with many markers. Here we attempt to estimate the effects of approximately 50,000 marker haplotypes simultaneously from a limited number of phenotypic records. And so here's really kind of talking about why they're looking at this, right? And this continues to be a major challenge with genomic prediction, right? Which is the N by P problem. We tend to have many more markers and genotypes than we have independent phenotypic records. And that has pretty significant implications on how you model the data. And now, of course, 20 some odd years later, we've largely perfected a lot of these methods. Well, I don't say perfected, but we've certainly gotten to a point where people are comfortable modeling this type of data. But at the time, this was a fairly new way of thinking about things. So again, the one thing I think to point out here with this background is that it does do a good job of hitting the key points of what needs to be covered in the background. But if you notice, they only took 2 sentences to do this, right? And that's really the key with the abstract, is how can you say something informative with as few words as possible, right? And when we're talking about the background, it really depends. This all ends up being a judgment call of the author, but you should try and do your background in 2 to 3 sentences. Here it's done in 2 sentences, and I think done quite effectively in 2 sentences. All right, so let's move on to the methods. A genome of 1,000 centimorgans was simulated with a marker spacing of 1 centimorgan. The markers surrounding every 1 centimorgan region were combined into marker haplotypes. Due to finite population size, effective population size equal to 100, the marker haplotypes were in linkage disequilibrium with the QTL located between the markers. again, what was done, the idea of methods is to make it clear what was done, keep it brief, and make sure the reader understands what was done. Now, there's a lot of details about this simulation that aren't included here. When I read the paper, I'll get that all from the methods section. But it is pretty clear to me that, this is a simulation paper. So they were using simulation. I have some idea about the genome that they simulated, about some of the parameters of this genome. It's quite clear that they were simulating a complex trait with many QTL associated with it, and that they simulated in a way to generate, good linkage disequilibrium between markers and the QTL. So it's, again, there's not a ton of detail here in the methods, but there's enough detail for me to understand what they did, and for me to make a judgment call of whether or not, what was done here really is relevant to whatever research I'm doing or whatever type of search through the literature I'm doing right now. So again, I think a good job here. The results section, the results section is one of the most important sections of an abstract. Using least squared, all haplotype effects could not be estimated simultaneously. When only the biggest effects were included, they were overestimated, and the accuracy of predicting genetic values of the offspring of recorded animals was only 0.32. So this is actually a pretty key finding at the time. In the 90s, there was a lot of focus on marker-assisted selection, where you would estimate the effects of markers, tracking major QTL, and you would use that to make selection decisions. Of course, we know that for kind of major gene effects, that approach works. At this point in time, back in the early 2000s, it was starting to become pretty clear that for complex traits that just wasn't going to work. And here they're kind of highlighting this idea of trying to go through and find major effect markers and only use major effect markers. It is not ideal for this type of complex trait. So that's a key finding there. Best linear unbiased prediction of haplotype effects, assuming equal variance associated with each one centimorgan chromosomal segment, yielded a much better accuracy. And they make a note that this was true even though the assumptions of the model were far from true. And that's good to know, right? So this is essentially a ridge regression bluff that they ran here. And it's clear that it did a pretty good job, even though the assumptions of QTL kind of evenly spread across the genome, all with more or less the same impact on the trait. Even when we make those assumptions and they're not true, it still does a reasonably good job of prediction. And then finally, Bayesian methods that assumed a prior distribution of the variance associated with each chromosomal segment increased this accuracy to 0.85, even when the prior was not corrected. So here they're kind of going a bit further. They're looking at some Bayesian methods that try and make assumptions about the distribution of marker effects that are maybe a little bit more closer to what we might expect, from our biological knowledge, and we did get some improvement in the simulation here. So again, results section pretty good. Then finally, we'll move on to conclusions. What were the main conclusions of the research? It was concluded that selection on genetic values predicted from markers could substantially increase the rate of genetic gains in animals and plants, especially if combined with reproductive techniques to shorten generation interval, right? So this is essentially the strategy of genomic selection. So this kind of, there were other papers that came out around this time that were sort of touching on similar concepts, but this is a paper that kind of kicked off the whole concept of genomic selection, which is now becoming quite common in plant breeding. It's been common in animal breeding for some time now. But again, a great conclusion written in a single sentence, right? So it is possible to convey a lot of information in a very limited amount of space in terms of the number of words you can have in an abstract. But you have to really think about what you're doing, and you have to put a lot of thought into how you're doing this. One thing that was, I guess a little eye-opening for me is after kind of sort of refreshing myself and looking at, what makes a good abstract. I started going through, obviously, and reading a lot of abstracts, and it was surprising, both in abstracts that I've written and abstracts for my papers, and just sort of generally kind of looking through the literature. There are a lot of abstracts out there that really don't follow kind of these best practices. And it seems quite common to have abstracts that really don't do a good job on hitting these four components, right? And so, you know, if you're someone that's struggling to write, you know, really effective abstract, you're not alone. It seems like, you know, the majority of scientists do, you know, struggle with this. So what are some of the common mistakes, right? So probably the most common thing, at least in some of the abstracts that are on the papers I'm an author or co-author on, not providing a complete summary and not paraphrasing your work, right? And so I think there is a bit of a temptation sometimes, especially if you're writing an abstract for a research paper, to go through and just say, okay, I'm going to grab a sentence from my introduction. I'll grab some sentences from my methods, some sentences from my results, and some sentences for my, you know, discussion and conclusion sections, and I'm going to try and kind of mash them all together. That really doesn't work. You should not be, you should really approach the abstract as being completely independent of the paper, right? So in other words, you've written your research paper, and now you're going to write the abstract, and you should not be taking sentences from your paper and trying to sort of cut and paste them into the abstract. You know, the problem with that's going to be that you know, when you're writing the paper and you have you know, a lot of space to write out, you're going to write your sentences, and you're going to structure, you know, how you're writing that very differently than the way you need to structure it from the abstract. So you really need to start from scratch when you're writing an abstract, including too much or too little detail for the main components of the abstract. And then, you know, this is by far and away probably the hardest thing to do in an abstract, right? Is we need to hit these 4 points. And we need to do a good job of hitting those 4 points, right? The background, the methods, the results, and the conclusions. And that means, you know, you're going to have to make a lot of judgment calls and how you're going to balance, you know, the amount of detail you put into each of these components. And it's just a hard thing to do. And it's something that takes, you know, to be honest, a lot of practice to get good at. You shouldn't be including citations. You should try and avoid abbreviations as much as possible. You know, that's not always possible. And, you know, if there are abbreviations that are very common and well known in the field, then, you know, those are certainly fine. And you know, detailed measurements. You know, you know, you have lots of room in the actual paper, or, you know, whatever presentation you're going to give like a lot of really detailed measurements and results. Again, the purpose of the abstract is really just to you know, inform, you know, the reader what is in this paper, so they can make a judgment call of whether or not they're going to invest the time in actually reading the paper, or they're going to invest the time in coming to, you know, coming to your poster, coming to your presentation. Another common thing is not really explaining what your results mean. I've seen a lot of abstracts where, you know, they'll say what they did, they'll put a lot of results out, and then, you know, there's no real kind of conclusions there to say, okay, this is why these results are important. They just sort of leave it to the reader to you know, make those inferences, and that's you know, an assumption you shouldn't make when writing an abstract. And then this one's another important one, not including keywords, or not putting enough thought into providing keywords. And down here I do have the source, so you can follow this link, and this actually I think is 11 common mistakes. I'm sort of condensing and paraphrasing what was in this this blog post here, but this is certainly worth a look as well, right? And this is something that I must admit, in the past I have not put a lot of as much thought into keywords as I probably should have, but the keywords and how you use keywords in both your title and your abstract is important, because it's going to determine what priority, what sort of relevance a search engine is going to give to your article, right? So let's go back to this sort of fairly generic, and then to be quite honest, it's not a very good search. Genomic selection in maize, right? If you look here, this search, because it's so broad and kind of vague, brought back 266 pages of results. Now, a lot of these results probably aren't very relevant to what I'm looking at. I will say that there's no way I would ever have time to go through 266 pages of results. And so, one or two things are going to happen, right? Like either, some papers that are of interest are going to, show up right here on this first page for me to find. And those are going to be the ones that I'm more likely to look at. More likely what I'm going to do is I'm going to try and, refine my search criteria to get down to something that's a little bit more reasonable. But as someone that's writing an abstract, you really do need to think about, who do you want to read this paper, right? So who is kind of your target audience, and think about, if you were going to some search engine, Google Scholar, PubMed, whatever it may be, and you were going to start searching for an article that's, covering some of the topics you are, like, what would be the keywords that you would type into the search engine to find it? And the more you can use those keywords in context, in your title and in your abstract, the more likely it is that your article is going to show up here on some of these front pages. Just so happens that with this keyword search, one of my papers ended up being on the first page because Sekiru, obviously did a pretty good job of thinking about what keywords he put into his abstract. All right. So now we've talked a little bit about, what, at least in concept makes an effective abstract. We've talked a little bit about some of the mistakes that are common. I thought it might be good to go through a few of the old abstracts written by me. And the point here is to be somewhat critical of these abstracts, so we can kind of look at, what was done well and what was done wrong with these abstracts. And I think some of the, abstracts that I wrote as a student is a good place to start as anything else. All right. So the first abstract I'm going to go through, and I'm going to kind of, from my perspective, kind of looking back at this abstract now, 20 some odd years later, and kind of give my thoughts of, what was good or maybe not so good about these abstracts. The second abstract that I'm going to cover, it'd be nice to get thoughts from the audience here, either in chat or feel free to unmute or raise your hand. It'd be hard for me, I think, to fully keep up with chat while I'm presenting, but feel free to interrupt me at any point in time. All right, so let's start with my first published abstract. That seems like a good place to start. This is obviously at this point in time, I was I don't know, I was probably a second year master's student. So I didn't have a whole lot of experience writing abstracts at this point in time. So I guess it wouldn't be surprising if this wasn't the best abstract in the world, but let's go ahead and let's walk through it. All right. All right, so let's start with the background. What is the subject of the research and why is it important? All right, so here's my background statement. Genetic evaluation of growth in Gelbvieh beef cattle was examined by multiple trait and random regression analysis. All right, that's it. That was my big background to let everyone know, what I did and why it was important. Now, the first thing that really jumps out at me here is the subject's clear. I was looking at growth in beef cattle. I was using multi-trait models, and I was using random regression models. So the subject's clear, but it's not clear really why is it important. Like, why am I comparing multiple trait models to random regression models? Now, there is a very clear reason why this was being done, but that's not stated anywhere explicitly in this abstract. So I think, unless you happen to already know what the limitations of using a multi-trait model for evaluating growth were, it wouldn't be obvious at all why we were looking to shift away from multi-trait genetic evaluations to random regressions. And the problem, really here is that, growth is a continuous longitudinal trait, and that's constantly changing through time. Multiple trait model it was essentially modeling snapshots in time and making very strong assumptions about, how the variance and covariance between measurements at different time points change. Random regression models provide a much more flexible framework to do it. And it actually was a big problem with the evaluations at the time because there were a lot of records that just had to be thrown away because of the sort of rigid nature of the multi-trait model. Now, to say that in an abstract wouldn't work, right? That was way too wordy, but, clearly, I did not do a good job on the background. So I think we can probably leave it at that now. All right, so now let's move on to the methods. This is, I'm not going to read through this method section, but it is very detailed. Again, providing some detail is not necessarily a bad thing. You want your abstract to be as detailed as possible, but there, there is, well, for one, there's abbreviations everywhere in this abstract. And I would say these are not necessarily standard, widely understood abbreviations. So that's, a mistake that I try to avoid now. But at the time, and it's tempting to use abbreviations a lot in abstracts, right? Because it enables you to get a lot more information into the abstract. But it's definitely something you need to be careful about. But the real question here with this method section is, is all of this information really needed, right? So I talk about what's in the data set. That's pretty important. But I also provide a lot of probably unnecessary detail about both the random regression models and the multi-trait models, right? So if someone is looking at this abstract and they have no idea what a random regression model is, or they have no idea what a multi-trait model is probably not a paper that's going to be particularly informative to them. So I don't think that, burning a whole lot of, my space in this abstract of providing kind of, really detailed information about some of these models is not a good use of the space here. All right, results. The results actually in this case, I think are pretty good, right? So the random regression models with the Legendre polynomials did not reach convergence until diagonalization. After that, it was found that all longitudinal models required few iterations to converge in the multi-trait model. I go on to provide some correlations, between the performance of the multiple trait models and the random regression models. I don't go into a lot of details about these, but I do provide some numbers. But in general, this results section is not so bad. If I was to go back and rewrite this, I probably wouldn't do a whole lot with the results section. I'm pretty satisfied with how that was put together here. And then again, the conclusions, the lower correlations for weaning weight and yearling weight indicated that the added information does affect breeding value prediction. The random regression model has the capability to incorporate records measured at all ages into genetic evaluations at a computing cost similar to multi-trait model. Now, again, this conclusion isn't too bad. I mean, it's actually a conclusion statement that I'm pretty happy with. Going back and reading this now, 20 years later. But I think the significance, particularly of this first statement, would be a lot more apparent to the reader if I hadn't done such a terrible job with my background statement, right? Because I never really stated clearly why we were even doing this sort of finding about the random regression models probably doesn't seem as impactful as it should seem, right? Because this actually is a pretty big deal. And, it changed the way a lot of the genetic evaluations have been done in beef cattle, but that wouldn't be very clear based on how I wrote that introduction.
Dr. Robbins,
yeah.
Before you move to the next slide, I think one person is asking what will be like an alternative background to that abstract.
Yeah. So I would say that there's nothing inherently wrong with this sentence right here, right? So the genetic evaluation of growth in Gelbvieh cattle was examined by multiple trait model and random regression model analysis. So I think that sentence can stay the way it is because it does clearly state what we did. What's missing here is there needs to be a second sentence that says why, we were comparing multiple trait models to random regression models. Something along the lines of, due to limitations in how multiple trait models handle covariance between phenotypic measurements at different times, significant amounts of data often have to be excluded from the analysis. Random regression models provide the ability to, provide the ability to have a framework that allows for continuous modeling of covariance and therefore including all available information for genetic evaluations. Now, again, that sentence was way too long and rambling. If I were to redo this, I would probably, start by writing out this second sentence that says why, and then I would probably spend a good amount of time trying to figure out how to paraphrase and cut that sentence down to make it as compact and efficient as possible. And that may take some time, right? So, and that's something that you really need to think about. Getting a good abstract takes time because it is a lot easier to, provide all of the necessary information when you have, essentially unlimited space to write. But when you're, forced to do it in a very, limited, amount of space in terms of number of words, let's say an average abstract might be around 250 words, right? You have to really think about how you say something. So that would be the big change I'd make is this background clearly needs a second sentence. Of course, if I add a second sentence to this background to cover the why, not just the what, then I'm going to have to start cutting from other parts of the abstract, right? Because that's going to put me over my word limit now. So the second thing I would do is I would start to look through, in particular, this method section here, and try and figure out like what really needs to be here, right? Like does the fact that my spline function, had the knots at day 1, day 205, and day 365, is that really crucial information to have in the abstract? No, it's not. I mean, knowing this is not gonna have any impact on whether or not this is a paper that I think I need to read. It's completely unnecessary, right? So this would definitely go. And again, explicitly defining like what the ranges, for data to be included in the multi-trait model are, again, I don't think that's needed. I mean, I'm using up a lot of space here to list out these numbers, but the reader doesn't need to know that in the abstract. So that can go too. And then finally, this statement here, although the results I thought overall were pretty good, the fact that the Legendre polynomial required diagonalization to get it to converge, again, I don't think that's really important. I mean, what's important is that, when we did do this process, it worked quite well. It proved to be superior to the multi-trait model. And this little tidbit of information obviously needs to be in the research paper because it is a result that people need to understand, but it doesn't need to be in the abstract. So I would definitely cut that out as well. And so I guess in kind of short, I would start cutting from my results and my methods to make room for me to actually write a proper background. So that's what I would have done here. And again, I would have to put a lot of thought into what that second sentence would be. And I'd probably have to redo this first sentence a little bit as well too, just to try and get down to a couple of relatively short sentences that say what need to be said. All right, so now we're going to jump ahead several abstracts later. Now, one would assume that at this point, this would have been getting towards maybe I, 4 years into grad school at this point. I've done my master's. I'm a decent way through my PhD at this point. I think one would assume that I've probably gotten better at writing abstracts at this point in time, but let's take a look at it, and I'd be interested to get some feedback from some people here. All right, so let's start with the background. What is the subject of the research and why is it important? I've clearly swung in a different direction on this abstract. I went from a one-sentence background that didn't have a whole lot of information into it to this. All right, so the use of gene expression data to diagnose complex diseases represents an exciting area of medicine. However, such data sets are often noisy, requiring the selection of feature subsets to obtain maximum classification accuracy. Due to the high dimensions of many expression data sets, filter-based methods are commonly used, but often yield inconsistent results. Optimization algorithms can outperform filter methods, but often require pre-selection of features to achieve good results. To address the problem of many commonly used feature selection methods, the ant colony algorithm is proposed for use on data sets with large numbers of features. The ant colony algorithm is an optimization algorithm capable of incorporating prior information, allowing it to search the sample space more efficiently than other optimization methods. All right. Now I want to pause for a minute, and I want to see if anyone has some comments on this background. Good, bad. If I were to rewrite this abstract, would I want to think about making some changes here? You can post in chat or unmute. I'd really like to hear what some people think about this. I wrote this abstract a long time ago. You're not going to hurt my feelings, so don't worry about it.
Okay, feel free to unmute yourself or put in the chat what you think about the background for this abstract. So personally, I will give my thoughts on this. Yes, like the person in the chat, I think the intro is kind of very long. We could maybe combine or abridge some of the reasons why it's important, and then just go directly to the next section. So you have a couple of responses as well.
Yeah. Too long. Yes, this is way too long, right? And I'll tell you what probably happened here, right? I probably wrote like a really good introduction in my paper, at least what I thought was a really good introduction in my paper, and I just couldn't let that go. And I probably took a lot of the intro of my paper and just put it into the abstract because for whatever reason, I thought I had written a great introduction. But yeah, this is way too long, right? Like I spend over half of this abstract on the background. And yeah, so I now have less than half of the abstract to cover all 3 additional sections, right? Like this is just way too long. All right, so now let's move on. Methods. Any comments on the methods section there?
I think a little more detail on the methods.
Yeah, the methods aren't.
Yeah.
It's debatable whether or not they even have methods in this abstract, right? Like this is not even a full sentence. And part of the reason why there's no detail here is because I spent so much space writing my background. I don't have any space left to actually talk about methods, right? So it was applied to several high-dimensional data sets. Like, what is that? I mean, like, what kind of data sets were they? What do I mean by high-dimensional, right? Like, there's no way that anyone reading this abstract is going to have any real sense of what I actually did. Like, what was I running the ant colony algorithm on? What was I comparing it to? What type of data was I using? What do I mean by high-dimensional, right? So yeah, this is, yeah. not good, not good. All right, so let's move on. Results. The ACA was able to identify small subsets of highly predictive and biologically relevant genes without the need for extensive pre-selection of features. Using the selected genes to train a latent variable model yielded substantial increases in prediction accuracy when compared to several rank-based methods and results obtained in previous studies. Any thoughts here? Yeah, and just throw anything out. I mean, there's nothing, right or wrong.
Yeah, I think it's the result is it's summarized enough, passes the message.
Yeah, of all the things wrong with this abstract, the results probably is, doesn't rise to the top of the list, right? There again, there is some detail lacking here, right? Like I think, having, providing some idea of what the added benefit was of using like an optimized searching algorithm as opposed to just, doing something like the t-test and then sorting, right? But it is clear that, that the ACA worked well, but, if I had actually provided some details on the methods, people could probably put this in better context. The fact that I didn't really have a methods section doesn't help with that. Yeah, and now let's go on to the conclusions. The superiority of the ACA algorithm was validated through simulation. I mean, that's not really even a conclusion. It's kind of somewhere in between a conclusion and just a continuation of the results section. So I'm not even sure this abstract really has a clear
take-home message.
Yeah, there's no real clear. I mean, the take-home message, I guess, is that it comes through a little bit in the results, right? That rank-based methods are not a good way of doing feature selection. And that does apply to work that we see now, where I think people look at using like association mapping or something like that to kind of decide what goes into a prediction algorithm, but in general, those types of rank-based methods aren't a very good way to do that. Yeah, so this is actually a pretty bad abstract. In my opinion, right? Like, it was a little bit painful for me to go back and read this. I mean, it's a good paper. It's actually a pretty well-cited paper. So, for whatever reason, despite there being a bad abstract, some people, ended up reading it and citing the paper. I'm actually quite proud of this paper. But the abstract is just terrible. It's just a bad abstract. And again, this bad abstract was really a result of me not really appreciating the importance of an abstract, right? Like, I'd written my paper. In my mind, the paper was the important thing. The abstract was just some kind of annoying little thing that I had to do. And I guarantee you'll probably find several of these sentences verbatim in my paper, because I guarantee you what I did is I went in, I picked out sentences that I liked in my paper, and I cut them and pasted them into my abstract. I probably did some minor editing to get into the word count, but I made that sort of really fundamental, really bad mistake of not making my abstract a completely independent and complete summary of my work, and not doing a good job of paraphrasing what was in the paper, right? You should never be cutting and pasting sentences from a paper into the abstract. They need to be paraphrased, which means that they need to be much shorter summaries of what you have in the paper. You're gonna have to rearrange things, and you really do have to start from scratch when you're writing an abstract. All right, so that was a little bit depressing to go back and see how bad that abstract was in particular. But
You have some
But
comments in the chat if you want to take a look at those.
yeah, no, yeah,
Okay.
I think that all of these comments are pretty spot on, right? I agree 100% with what's coming in on the chat here. And yeah, and I will say that this is, you know, actually going through and sort of dividing an abstract up into its component parts, I think makes it easy to recognize pretty clearly what needs to improve. And yeah, you know, the fundamental flaw with that abstract was I spent so much space on my intro that I really couldn't do justice to the methods or the results or the conclusions, right? The real problem with that abstract was that the background was way too long. By shortening down that background to maybe 2 or 3 sentences, I would have a lot more space to fix all of the problems with the methods in particular and the results and conclusions. All right, so now let's move on to an abstract. I've graduated with my PhD. I'm working in industry now. At this point, I've gotten pretty efficient at writing papers and abstracts. I don't know how many abstracts I would have written and submitted at this point, but too many to count. And let's see where I'm at now, right? All right. This is some work I did on the use of two-dimensional splines for doing spatial corrections of field trials, right? For trials exhibiting within-field spatial variability, good predictions of genetic merit require models that accurately estimate and partition spatial effects from genetic sources of variation. Ideally, such models should be robust, providing good solutions across a range of spatial patterns. To achieve this, a new method for spatial corrections is proposed, which utilizes two-dimensional spline interpolation to estimate spatial effects. Okay, so, you know, this is probably still a little bit on the long-winded side. This could probably be tightened up a little bit, but again, I pretty clearly state, you know, why we're doing what we're doing. And then, you know, I pretty clearly state kind of what we did in this paper, right? So if you read this intro, you're aware that, you know, finding robust and accurate ways to do, you know, spatial corrections on field trials is what we're looking for. And specifically what we did in this paper is we proposed, you know, at this point in time, probably autoregressive models were the standard. We're proposing a different way of doing this using two-dimensional splines. So yeah, so not a, yeah, I mean, you know, everything can be improved, but not a bad background in my opinion. Methods. Simulation experiments were used to compare this method with two-dimensional separable autoregressive models and mixed models containing marginal pass and range effects. Models were evaluated based on the accuracy in partitioning genetic and spatial sources of variation, as well as robustness across different spatial and genetic effect structures. All right, so again, you know, it's clear we're using simulation here for better or worse. It's clear what models I'm comparing the two-dimensional spline to, and it's generally pretty clear what criteria I'm using to evaluate these models. You know, obviously we want to be able to separate out genetic and spatial sources of variation, and we want to find something that's pretty robust. In other words, it's going to, you know, work well across a variety of scenarios. And so I don't go into detail what scenarios we simulated. I don't think that's necessary in an abstract. I don't think you could really even do that in an abstract. But I am pretty clear that we looked across a range of scenarios for spatial and genetic structures. All right, and then finally, results showed the two-dimensional spline method to be the most robust of the 3 methods, yielding accurate spatial corrections across all simulation scenarios, whereas the autoregressive model showed indications of overfitting in the presence of genotype by environment interactions. And the pass range model performed poorly in the presence of pass by range interactions, which is not surprising because that model doesn't account for them. Applications using maize, hybrid yield data, and moisture data confirm the effectiveness of the two-dimensional spline at reducing air variance by accounting for spatial trends. So again, I'm not providing any kind of detailed, measurements of accuracy, but I am, I am giving a reasonable summary of what the results were. I think, if you read the paper, you might come back to this abstract and say, well, maybe there's a little bit of bias creeping in there because, there were some limitations to the two-dimensional spline. There were actually a lot of scenarios where the autoregressive model was better. And so I probably should have given a more balanced accounting for the results here than I did. And then finally, this new model provides breeders with a robust method to model within field spatial variability across a range of experimental designs and spatial effect distribution. I think that's actually a pretty good conclusion. I think that kind of summarizes, what the potential impact of this work is. So this is I venture to say, you could probably find something wrong in just about every abstract, but this abstract, hits the four main components of the abstract, and it, more or less does a pretty reasonable job with each of those components. So I would say this is definitely a vast improvement over the previous abstract. I think this is probably, a bit more representative of what an abstract should look like. All right. Some comments. How do you assess the impact of a paper from its abstract? Most times I have the Earth's paper, but I don't know where to start from. All right. Let me see. There's some. Okay, so it looks like above that it's more or less comments. All right. So, the abstract isn't necessarily about conveying the impact of a paper. Ultimately, the scientific community is going to be the judge of how impactful that's going to be, and that's going to depend a lot on how many people actually read your paper and how many people use those results to inform their own research. That's where the impact comes in. The purpose of the abstract is to make sure that any researcher that comes across your paper can read the abstract and can make an informed decision about whether or not, regardless of how impactful your research is, right? Like, you could have a research paper that's incredibly impactful, but if it's an area of research that I'm not involved in, then I'm probably not going to read it, and I probably don't need to read it, right? Because it's far enough outside of my field, it's far enough outside of where I'm working that other than maybe just curiosity, it's probably not that important for me to read. The worst thing that can happen, right, is for someone to read your abstract, not really have a good idea of what you did, and say, well, I don't really know what this is, and based off this abstract, it's not clear to me that this is really useful for me, and then I move on, when in fact the paper itself, if I had read it, would have been relevant to me, would have potentially impacted my research. That's like the worst possible thing that can happen. And so again, the abstract just needs to be a complete independent summary of your work. It needs to be clear, it needs to be succinct, and you need to make sure that someone that reads this abstract is going to be able to make an informed decision about whether or not your paper is relevant to their research or not. And that's really all you're going to get out of an abstract. And that really should be your sole focus of an abstract. As far as the second question, that's probably a topic for a whole nother seminar, right? How to start when you're writing a research paper. But generally what I like to do when I'm writing a research paper is the first thing I do is I list down, what I think are the key results from the research and what I think are kind of like the key conclusions or the key takeaways from that research. So I start by sort of listing that out and saying, okay, here are the 3, 4, 5 points that I want to really clearly make in my paper, because these are sort of the takeaways that I want people to have from this research. Once I have that down, then, typically I'll go back and I'll say, okay, well, what results, do I need to include in here to support this? How should I be, presenting these results? And that's always a bit of a trial and error process. But the key thing, before I start doing anything else is, again, to kind of list what I think are the key results, list what I think are the key conclusions and takeaways. And then from there, I start thinking about, okay, well, how am I going to structure this paper in terms of how I deliver results, sort of the order in which I talk about things, to sort of build, a narrative that makes it very clear, what was done, what the results were, and why those results are important, and what the real takeaways are. And papers that do a very good job of sort of laying out a very clear kind of narrative, kind of walking through the paper in an order that kind of builds upon itself and builds towards significant conclusions, are going to be papers that I think are going to be more well read and cited better. One thing, again, similar to kind of like with an abstract, sometimes what you see in papers is people present a lot, right? So you've spent however long working on this project, you probably generated a lot of data, right? There's probably a temptation to put everything in there and talk about everything, right? But again, that's a temptation that you have to be careful with, right? Because if you talk about everything and you try and explain everything, right, sometimes the main points can get lost. And people kind of go away from reading the paper, a little bit confused and not necessarily sort of sure what kind of the important points were, what was, kind of minor things that were maybe, not sort of central to the purpose of the research. But again, that's kind of a long-winded answer. I think the short answer is you really just need to, start by, again, what are the key points you want to make? What are the key results? And then I always start to frame out like an outline. I start to frame out sort of what I want to talk about when, and then I iteratively go in and fill it in. The abstract should be the last thing you write. And usually even like the introduction is something that I write towards the end as well. Because I want kind of the results and discussion to essentially drive the introduction. Methods, methods you can kind of write anytime, I guess. And then, yeah, definitely the abstract last. All right. So again, writing an abstract, you can be a great writer and still write really bad abstracts, right? Because again, with an abstract, it's not about, how well can you put together a sentence, how good are you at, how good is your command of the English language if you're submitting to, maybe a conference or a journal that has their abstracts in English. Those things are important, right? But the added thing with an abstract is that skill of being able to convey a message very concisely. It's informative and representative of your paper, and that takes a lot of practice. And so, if improving on abstracts is something that you personally feel needs to happen, the best way to do it is to practice. And so, I would recommend, if you have some experience writing abstracts, so you've written abstracts in the past, the first thing I'd do is, again, I would maybe go to, some of these online resources that will kind of say more or less what I said in this topic, in this presentation, because I drew a lot of this material from those online resources, but be there for you as a quick reference. But I would go through and I would review your abstracts. I would kind of, repeat the exercise I did of seeing, like, can I actually clearly pick out these 4 components, right? And then using some of these tips from this presentation or online or wherever you can find a good source of tips for writing an abstract, I would try and rewrite those abstracts to improve upon them, right? I didn't actually do that because I didn't have time. I guess I would have liked to have gone back and rewritten some of those abstracts, in a different way, but unfortunately didn't have the time. But I certainly encourage people, attending here that want to do that, to definitely go through and rewrite some of those abstracts. If you don't have any experience writing abstracts, there was a tip that came from this article here that I thought was really good. Pick a scientific article in your field, read the paper with the abstract covered, right? So just take an article that's in your field, completely ignore the abstract, don't look at it at all, read the paper, then try and write your own abstract for that paper based off what you're reading. And then after you've written your abstract for that paper, you can start to compare it to the authors. And this is a process that you can repeat until you feel like you're doing a pretty good job of summarizing, a paper. I mean, when you're doing this, keep in mind, my abstract with the ant colony algorithm is a prime example, right? Not all published abstracts are good. Keep that in mind. But in general, if you can go through and write an abstract that's picking up on some of the same points that the authors put in their abstract, you're probably getting to a point where you're writing pretty good abstracts. And so again, practice makes perfect. No one's going to be good at writing abstracts the first time. They just aren't. So the only way you're going to get really good at writing abstracts is to practice. So. And with that, that's all I had. I'm happy to answer any questions. If I missed anything in the chat, please let me know. And again, thanks for having me here. And I really appreciate the invitation, the opportunity to meet everyone and give the talk.